without further ado, if we wanted to get started, first up we have Elicia Cabrera in the Pay Lab. If you wanted to go ahead and share your screen. You're on mute if you're, yeah.
Do you see the presentation?
Yes.
Okay. Hello, I am Elicia Cabrera, a rising junior at Florida Agricultural and Mechanical University, studying biochemistry. I had the honor to be accepted into the HuBMAP summer internship program. My presentation will be going over single-cell multiomics with human heart tissue. This was my very first internship experience, and I found it very valuable and insightful. HuBMAP is the Human Biomolecular Atlas program, and the goal of HuBMAP is to create a single-cell resolution, multi-scale spatial atlas of the healthy human body. You guys see the next slide?
I can't see the next slide yet.
Okay, here you go. Can you see it now?
Yes.
Okay. I was having trouble. Single-cell multiomics is a technique for analyzing multiple types of molecules from individual cells. The key components of single-cell multiomics are single-cell isolation, which is separating individual cells from a sample, barcoding, which is assigning unique identifiers to each cell for tracking, and sequencing, which is determining the sequences of DNA, RNA, or proteins within each cell. It can be applied for cell type identification, which is classifying different types of cells based on their molecular profile. Functional analysis, which is understanding the roles and functions of specific cells, and pathophysiological insights, which is studying cellular processes in health and disease. It has great impacts on research for enhancing our understanding of cellular diversity and function, advancing personalized medicine by identifying specific cellular targets, and it contributes to the creation of detailed cellular atlases for various tissues and organs. This summer, I was in Pay Lab at Children's Hospital of Philadelphia. We collaborate with four other labs, each holding a crucial role. Children's Hospital of Philadelphia is a tissue mapping center, but is also part of the demonstration projects for HuBMAP. The objective of our lab is to process the human heart samples and to conduct the single-cell multiomics. This involves intricate steps in sample preparation and analysis. Our goal is to contribute to the creation of the comprehensive heart atlas. This atlas will provide valuable insights into the heart function and advancing our understanding of therapeutic strategies. Okay, how we collect. We follow a meticulous process to collect the heart samples. We start by obtaining informed consent from the donors or their families, and then we ensure the inclusion of diverse populations, as this allows for a comprehensive understanding of heart health across different demographics. Our sample collection methods are rigorous but consistent, ensuring high quality and reliable data. Why we collect. The hearts we collect come from individuals who were organ donors, but unfortunately were not matched to a recipient. We honor their donation and contribute to vital research by collecting these hearts. The collected hearts are compared to historic data in our extensive heart bank, which contains over 1,200 samples. The comparison helps us to identify patterns and anomalies that can lead to breakthroughs in cardiac research. After meeting all the criteria, the heart can be collected. During my internship, I had the opportunity to be present for two heart collections. Our lab collected samples from the left and right atrium, left and right ventricles, AV node, and the SA node. Once the sections were collected, we subjected them to retrograde perfusion with cold cardioplasia, and then they will be frozen or fixed within 4 hours to preserve their integrity. I participated by minting the heart sections before they were stored in liquid nitrogen. To start this intricate process, we had to carefully harvest the nuclei from the frozen heart tissue samples. The first step involved chopping the heart tissues into fine pieces using a blade and forceps. This delicate procedure ensured that the tissue is adequately prepared for the next steps. Once finely chopped, the tissue pieces were added to the test tubes, including a lysis buffer. This buffer helps to break down the cell membranes, releasing the nuclei. After the lysis steps, the samples underwent many washes and purifications to make sure that the nuclei were good. The steps are crucial to remove any remaining cell debris, ensuring that we have a clean sample for further analysis. After this purification, the nuclei were stained with a blue staining agent, which made them visible under a microscope. Observation under a microscope is a crucial part of the process as it allows us to count the nuclei accurately. Ensuring an accurate count is essential for the next stages of single-cell analysis. This comprehensive approach enables us to gain a deeper understanding of the cellular composition of the heart, as well as the functional roles of different cell types in heart and disease. After we harvest the nuclei, the single-cell molting process can begin. We begin with the transposition. This step allows to help us to open the nuclei, making the DNA accessible for further processing. After this is completed, we can move on to the gem generation and barcoding. Each nucleus is encapsulated into a gel bead emulsion and uniquely barcoded, allowing individual cells for tracking. During the binding, we get both the gene expression and the open chromatin region. TN5 combines with the open chromatin region, and then we can transpose from there. TN5 simultaneously fragments DNA, inserts into the open chromatin sites, and adds sequencing primers. This sequenced DNA identifies the open chromatin, and the DNA analysis can provide insight into gene regulation. For the gene expression, poly-DT is used, and it detects the poly region of the RNA. This figure shows a HuBMAP, which displays all the nuclei and the cell types we collected from 3 different hearts. The data comes from both the gene expression and the ATAC-seq, allowing us to identify various cell types in the figure. We see different cell types, including fibroblasts, cardiomyocytes, macrophages, and 7 other cell types. From the 3 hearts, we were able to collect 83,297 nuclei. During my time here at Pace Lab, I had the opportunity to learn many basic lab techniques which significantly enhanced my understanding of molecular biology. One technique I was able to learn and gain subsequent experience in was PCR and genotyping. This process started with obtaining the mouse DNA through a procedure called tail digestion. We would carefully cut the tip of the mouse tail and then subject it to an overnight digestion process using a specialized buffer and proteinase K. This step was crucial as it allowed for the breakdown of the tissue, thereby releasing the DNA into the solution. After the digestion process was complete, the samples, were ready for the next step. To perform PCR, it was essential to prepare a reaction mix. This mix included the template DNA, primers specific to the gene of the interest, nucleotides, and the Taq polymerase enzyme, which is responsible for DNA synthesis. We would then add this reaction mix to the PCR tubes containing the samples. The PCR machine, also known as a thermocycler, was used, and this amplifies the DNA through repeated cycles of deminiaturization, annealing, and extension. Once PCR was completed, we proceeded to run the agarose gel to separate and visualize the amplified DNA fragments. This step involved by preparing the agarose gel, loading the PCR products into the gel wells, and applying an electric site to separate the DNA fragments based on size. By comparing the bands of the gel DNA to the latter, we could determine the genotype of each sample. This comprehensive process not only solidified my practical skills, but also deepened my theoretical understanding of genetic analysis techniques. I learned many things during this summer. I was able to learn about the HuBMAP project and assist, but I was also able to learn basic laboratory techniques as well. I learned PCR, genotyping, mouse dissection, organ collection, embedding human heart tissue, and cell culture. But also, by being a HuBMAP intern, I had the opportunity to attend some amazing web seminars this summer, including Becoming a Resilient Scientist series and Brown Bag Talks with amazing scientists. I would like to thank HuBMAP and the NIH for this amazing opportunity I received. I'd also like to thank Children's Hospital of Philadelphia and University of Pennsylvania. I would like to give some acknowledgments to my PI, Dr. Pei, and as well as my mentor, Pei-Ron, for all the help and knowledge that they helped me obtain this summer. I would also like to give a shout out to everyone else in my lab for being so generous with letting me watch them do their experiments and learn many other things. What I learned this summer will definitely contribute to making me a better scientist in the future. Thank you for watching my presentation. And if you have any questions, you can ask them now. You can also connect with me on LinkedIn at Elicia Cabrera.